So in our last lecture we studied about non-membranous organelles, and in this lecture we will be discussing about single membranous organelles and double membranous organelles. So first we will be discussing about single membranous organelles. Before we study single membranous organelles, you have to understand what is endomembranous system. So endomembranous system is a system in which single membranous organelles are connected with each other through a common function. Like all of these organelles have been connected with each other and they share a specific function that is connected with each other. So as you can see here, the first organelle that we will discuss about is going to be endoplasmic reticulum. Then after endoplasmic reticulum release a vesicle. That vesicle moves to the Golgi apparatus. So the next stop is the Golgi apparatus, or you can say the Golgi complex. After the Golgi complex modify that vesicle, that vesicle comes out from the other side of the Golgi apparatus. So now the vesicle has two options. Either it can stay in the cell and become a lysosome, or it can also be removed from the plasma membrane by budding off from the cell. So that's how these functions are connected with each other. So endomembrane system is the system in which these single membranous organelles are connected with each other in a function. So we will discuss all these steps one by one in a really great detail. This was kind of a bigger picture that we will be discussing in our single membranous organelles. So let's move on to our first stop, that is endoplasmic reticulum. Endoplasmic reticulum is like made up of small elongated closed sacs, and those sacs are called cisternae. Okay, cisternae combines together to form an extensive network or a system of membranes, and it is called endoplasmic reticulum. So it is connected with the outer nuclear membrane, and the next thing is that it is one of the most largest organelles, and it contains occupies most of the cytoplasm, and it contains two regions. The first is SER, that is smooth endoplasmic reticulum. The other is RER, that is rough endoplasmic reticulum. Smooth and rough endoplasmic reticulum has differences. Rough endoplasmic reticulum contains proteins, while smooth endoplasmic reticulum does not contain proteins. What does I mean by proteins? That is actually ribosomes. So RER have ribosomes on its surface. That's why it is also called factory of protein synthesis. Ribosomes are a factory of protein synthesis, and they are present on RER. So RER indirectly also produces proteins. So that's why protein synthesis and secretion is a function of RER. Well, SER does not contain any ribosome, so it does not produce any protein. As you can see, the ribosomes are present on the surface of RER, like kind of dots and bulges that you can see, and these bulges causes the surface of RER to be rough. That's why it is called rough, because it has a rough surface, while SER does not contain any ribosomes. That's why its surface is smooth, so it is called smooth endoplasmic reticulum. Next thing is that what is the function of SER? Like we know that RER contains ribosomes, so obviously its function is protein synthesis. What about SER? SER helps in lipid metabolism, and it helps in drug or toxin detoxification from the body. It also helps in calcium storage, and it also helps in impulse transmission, like nerve impulse is transmitted through SER. Smooth endoplasmic reticulum also help in impulse transmission. The other function, it could be like transport of material. Transport of material is also a function of SER and RER both. Like transport is like the general or common function, but SER is mostly involved in transport. If we next talk about products that are formed, then RER forms glycoproteins, membrane proteins, everything that is related to proteins, while SER contains products for phospholipids, steroid hormones, triglycerides, and these are all the products that are formed in SER. So memorize these names because they are really important. Well, what are the types of cells that are present in RER? Like RER is mostly abundant in pancreatic and plasma cells, because they can have to make protein molecules. Like enzyme is made up of protein, antibodies are made up of proteins. That's why they are more abundant in these kind of cells. While liver cells wants to detoxify, that's why SER is more abundant in liver cells, while steroid secreting cells also contains SCR because steroid hormones are made by smooth endoplasmic reticulum, and muscles also contains SCR because SCR stores calcium plus 2 ions. If we talk about other differences, is that RER is like directly connected to Golgi apparatus, while SCR has no direct route towards the Golgi apparatus. If we make other differences, that would be like RER is like more stable than SCR, while SCR is less stable than RER. The reason is simple, because RER contains ribosomes while SCR does not contain any kind of ribosomes, and RER also give like mechanical and mainly provide support to the endoplasmic reticulum. That's why RER is more stable while SCR is less stable. That's why transport is a common function of both of these, like RER and SCR both helps in transport, and RER and SCR both helps in making the whole organelle stable, and mechanical support is given by both. But RER is majorly involved in support and SCR is majorly involved in transport. I think you got my point. Like transport and mechanical support is a common function of both of these parts, but transport is major function of SCR and stability is major function of RER. I think you got my point what I'm trying to say here. Next thing, let's study about other examples and other things that you need to know about endoplasmic reticulum. First thing is that in skeletal or cardiac cells, you have a skeletal or cardiac muscle cells. You have a form of SCR, and the SCR in these muscle cells are called sarcoplasmic reticulum. What is it called? Sarcoplasmic reticulum. We will study it in detail in our muscle chapter because it is a really important topic. Because sarcoplasmic reticulum, the function of sarcoplasmic reticulum is that it stores calcium ions, storage of calcium ions. As it is storing calcium ions, that's why smooth endoplasmic reticulum is more abundant in muscles and cardiac cells. The next thing is that if many ribosomes are attached on a small like cisternae of RER, then it is called Ergastoplasm. What did I said? That if many ribosomes are attached, like ribosomes plus are attached on small cisternae of RER, then what should we call it? It is called Ergastoplasm. Simple, right? You have to know that what does Ergastoplasm means. Then if we say in nerve cells, like in nerve cells, the Ergastoplasm is called, in nerve cells, the Ergastoplasm is simply called as Nissl's granules. What do we say it? It is Nissl's granules. So these were some extra points that you need to memorize for endoplasmic reticulum, and these are the points that we will discuss in our future lectures. Like sarcoplasmic reticulum will be studied in the muscle chapter, and ergastoplasm and Nissl granules will be studied in the nervous system chapters. So let's move on to our next topic, that is about Golgi apparatus. So if we talk about Golgi apparatus, like Golgi apparatus is the next organelle that will form by the endoplasmic reticulum. As you can see, this is endoplasmic reticulum, and this is a vesicle that was formed from endoplasmic reticulum, and it budded off from the endoplasmic reticulum, and then it came towards the Golgi apparatus. The one thing that you need to know about endoplasmic reticulum is that approximately all the single membrane organelles are formed by endoplasmic reticulum. Like if you remove mitochondria and chloroplast, all other single membrane organelles are formed by endoplasmic reticulum. Endoplasmic reticulum will bud off these kind of vesicles that are made up of proteins and lipids and phospholipids, and these vesicles combine together to form different vesicles, just like Golgi apparatus. So Golgi apparatus is also made up of cisternae that was bodied off from endoplasmic reticulum. So if we talk about a single cisternae, like one cisternae, if we say we have a single cisternae, then it is called as a Golgi body, right? And if we say that we have a stack or a bundle of cisternae, let's say it is a stack of cisternae, then it is called as Golgi apparatus. But what about Golgi complex? Golgi complex is actually cisternaes, all the cisternaes that you have plus vesicles. So as you can see here, if we talk about only this part, then it is a Golgi body. If we talk about this part, then it is only Golgi apparatus. And if we include all the systems like the bodied of vesicles and the stack of cisternaes, all of it combined together to form a Golgi complex. So that was about the basic concepts of the Golgi apparatus. It is kind of a post quality control center, just like if you are in a factory or a mill, then there is a quality control place somewhere in there that they control if the product that is going from one place to another place is worth delivering or not. So Golgi apparatus is like in between center that helps cell into sorting out everything and shipping center in our cell. So Golgi apparatus have another name that is called Dictyosome. You can also say like Dictyosome is another name of Golgi apparatus. Dictyosome, sorry, Dictyosome. Dictyosome is actually the name of Golgi apparatus in plants. Like Dictyosome actually helps in plants to form the cell wall. As you know that in plant cells we don't have invagination of the cell walls, cell membrane, sorry. So when cell division occurs in animals, the cell membrane invaginates inward, while in plant cells the cell invagination does not occur. There forms a Fragmoplast in the center, and the Fragmoplast forms and then two cells are divided. We will discuss it at the end of the topic again if you want to, and that's why it is called Dictyosomes in plants. So next thing that you need to know about Golgi apparatus is that, as you saw that the vesicle was coming from endoplasmic reticulum, and then first thing is that it will start forming this structure, and the structure that is here, it is called the forming phase. Like Golgi apparatus starts forming for here, so it is called as forming phase, and the other end here it is called maturing phase because it has already been matured and the vesicles started from up towards downward. So this was the forming phase and this was the maturing phase. So the vesicle will move from forming to the maturing phase. So these are like really important, and most students make really big amount of mistakes between two of these phases. The reason is that because they contains different kind of functions and different names. So first thing is that forming phase is also called convex, like it has a convex shape, and if you talk about maturing phase, then it has a concave phase. And if you say like what is the phase of forming phase, then it is called the outer phase. It is like a bit confusing, like maturing phase should be the outer phase and forming phase should be the inner phase, but it is actually opposite. The forming phase is called the outer phase and the maturing phase is called the inner phase. The next difference that you need to know about is that forming phase is also called as cis phase, while maturing phase is called as trans phase. Like these are important differences between these. So I want you to like make mnemonics or memorize them. These are really important and most students mix them up a lot. So I like write them down in your notes, revise them again and again, and please don't like make any mistake about this difference in your IMAT exam, because they might be talking about, like they say, what is happening at the cis phase of the Golgi apparatus, or what happened at the inner phase of the Golgi apparatus. They can say anything. And if you talk about the forming phase, like forming phase is, as you can see, towards the nucleus, right? It is like pretty clear, I think, in the diagram. it is towards nucleus. But if you see towards maturing phase, then it is away from nucleus, right? As you can see, endoplasmic reticulum, and this is the nucleus. So forming phase is facing toward the nucleus and maturing phase is facing away from the nucleus. So that was the difference between forming phase and maturing phase. Please memorize this table so you don't have to make any mistakes in your final exam. If you talk about the functions, so functions of Golgi apparatus are really important, like they are like the gold mine of MCQs. So the first thing that you need to know is that the first function that Golgi apparatus has is the cell secretions. It is the main function of Golgi apparatus, that it forms cell secretions. Our major body cell secretions are secreted by Golgi apparatus. The second major function is that it causes the modification of different molecules in your body. The major modification is that it converts carbohydrates, carbs, into glycoconjugates. What are glycoconjugates? You know that conjugate is a molecule that is formed by two different kind of molecules, like lipoproteins, glycoproteins, lipoproteins. So glycoconjugates are the conjugates that contains glycogen in it or glucose in it. For example, glucoproteins, glycolipids, these are glycoconjugates. So the major function and the major modification that Golgi apparatus does is that it converts carbohydrates, or you can say glucose, into glucoconjugates. So this was the major function of Golgi apparatus. The other function is that it forms Fragmoplast. Just we talked about above, like what was a Dictyosome. Dictyosome was the name of Golgi apparatus in a plant cell, and what does Dictyosome does is that it forms Fragmoplast. So what is Fragmoplast and what is invagination? If we talk about animal cells, so animal cells invaginate from inside just like this. If it is an animal cell, then it will break from between, and then you have one cell and another cell. That's how mitosis occurs in an animal cell. But if you talk about a plant cell, then plant cell will have fragments in the center just like this. This is called Fragmoplast, and one cell will move towards left and one cell will towards right. So that's how cell division occurs in plants. So Fragmoplast is formed in plants cells to cause the cell division. So Fragmoplast is actually formed by the Golgi apparatus. So formation of Fragmoplast is one of the major function of Golgi apparatus. The next thing is about the formation of acrosomes. What is acrosome? You tell me. You studied it in high school, right? Acrosome is actually the head of sperm. If I create sperm like this is your sperm cells. And in sperm, this head portion that I have highlighted is actually acrosome. That causes the penetration into the egg cell. So the tip or the head of the sperm is called acrosome. The tip of the sperm cell is called acrosome, and acrosome is formed by spermatogenesis, and it is created by what? Yes, Golgi apparatus. Golgi apparatus helps in the formation of acrosome. So these were the major functions of Golgi apparatus. If we talk about the pathways of vesicles, it is really simple. As you can see, the first thing that is formed is a protein that is formed in ribosome in endoplasmic reticulum, or you can say rough ER, and then the vesicle that is formed in endoplasmic reticulum will be transported in the form of transport vesicle towards Golgi apparatus. Golgi apparatus will receive that transport vesicle, and then it will modify that transport vesicle. After Golgi apparatus has modified it, then it will become a Golgi vesicle. And now the Golgi vesicle has two different pathways, as you have seen here in the previous diagram. Now this is called as the vesicle, right? Golgi vesicle. Now this Golgi vesicle have two choices. Either it will be removed from out of the cell, right, or it can be converted into lysosome. So the formation of lysosome is actually a function of Golgi apparatus, smooth endoplasmic reticulum. So this was the total endomembrane system. The endomembrane system is not finished yet, but the formation of lysosome has been done. So this was about Golgi apparatus and endoplasmic reticulum. If we talk about the next topic, that is about lysosomes. Like lysosomes are really important because lysosomes are also called splitting bodies. Like they are like your cytoplasmic organelles, which are found in like most eukaryotic cells, except they are not present in red blood cells and other cells, like due to their morphology, but they are mostly present in all of the cells. It was discovered by Dictyosomes. The scientist name was Dictyosomes, like weird name, but yeah. So Dictyosomes actually discovered lysosomes. So they are roughly spherical in shape, kind of spherical, and they are single membranous, as you already know. And the major thing is that they have digestive enzymes, and the digestive enzymes are called hydrolytic enzymes. There are about 40 hydrolytic enzymes like carbohydrases, lipases, proteases, nucleases, so many types of enzymes, and there are approximately 40 hydrolytic enzymes. You have to know that in your cells there are about 40 hydrolytic enzymes, and you know that where these enzymes are formed. So these enzymes are actually formed in RER. So RER is the one that helps in the formation of these hydrolytic enzymes. So this was also an important point that I must mention here. So these enzymes mostly work in an acidic condition, and the pH is approximately 5. So there are types of lysosomes that you have to understand in the diagram here. There is a primary lysosome, as you can see here. This is the primary lysosome, and then you have secondary lysosome, and then at the end you have tertiary lysosomes. So there are 3 types of lysosomes. Understanding the types from the diagram is much easier than just studying the definitions or studying the text. So if we see here, what is primary lysosome? Like if the enzymes are budded off from the Golgi apparatus, or then they will become Golgi vesicles, right? And the Golgi vesicles are actually called then primary lysosome. So if the vesicles are budded off from Golgi apparatus, then the first thing that their name is going to be primary lysosome, and then after the primary lysosome they will become secondary lysosome. So how secondary lysosomes are formed? Secondary lysosomes are formed by the fusion of primary lysosomes with food vacuoles, and you can say they are known as phagosomes, like phagocytic food vacuole. So secondary lysosome is formed when a lysosome, primary lysosome, attaches with a food vacuole or any kind of cell that is present, and it attach with a phagosome or endosome, then it will form a secondary lysosome. So this was an example of secondary lysosome. Like if there is a phagosome or if there is an endosome, then primary lysosome will combine with that and it will form a secondary lysosome. What is tertiary lysosome? Tertiary lysosomes are also called residual bodies because they contains like undigested material after absorption of food into the cytoplasm. So tertiary lysosomes are like the waste material, or you can say residual material, so that are left after secondary lysosome. So this was all about primary, secondary and tertiary. If you know that what happens in unicellular and multicellular cells, they are different. In unicellular cells, they are removed outside the cell by exocytosis. I am talking about tertiary lysosomes. And in multicellular cells, they are like retained in the body and they form granules and they stay in the body for long terms, and they are eventually like detoxified. So this was like the primary, secondary and tertiary. Primary is budded from Golgi apparatus, then it will fuse with either a phagosome or endosome, it will form a secondary lysosome. We have discussed about phagosome and endosome in our topic for cell membrane, so you know what is phagosome and endosome is. And the next thing it is that after secondary lysosome will start digestion. It will digest the phagosome or endosome, and the remaining body is called as tertiary lysosome, and it will be either removed from the cell or it will stay in the cell. So this was the type of lysosomes. If we talk about functions, So the functions of lysosome are really important, because there are 4 different diagrams that you can see here, and they are 4 different major functions of lysosomes. First we talk about phagocytosis. So whenever a different organism, I'm saying organism, any foreign particle that comes into your cell, for example this was a foreign particles, they entered into cell through endocytosis and they became endosomes. Anything that came here, they become endosome. What does it do is that these endosome will attach with the lysosome. What does lysosome does is that lysosome will eat them up, so they cause phagocytosis. Phagocytosis eventually literally means eating up of anything that is present there. So phagocytosis means eating. You know what is the meaning of lysosome? Lyso means splitting and soma mean bodies. Like lysosome is literally called splitting bodies. Like split means breaking down anything. Like lysosome has only one function, and that is destruction. Like it is a war machine in your cell. It will destroy up anything, it will eat up anything that comes into your cell. So next function is about autophagy, about cell death and exocytosis. If we talk about autophagy is really important, because there are 2 type of functions. What is autophagy and other is autolysis. There is a difference between autophagy and autolysis. Like they are different, you have to know the difference between them. Autolysis. So what is autophagy and autolysis? Phagy means eating up, so autophagy means self-eating of the cell. For example, if a cell is not getting nutrition at all, if you are like in a place where there's no food and no water, what does cell starts doing? Lysosome will start eating its own organelles. Slowly it will start eating everything that is in its front of it. So lysosome will eat everything that is there in the cell, and it is called as autophagy. So it is also called as self-death, or you can also say it is kind of a suicide bomb. Lysosome is also a suicide bomb in your cell, because it can eat up everything that is present there. The other function is called autolysis. So autolysis is also similar to autophagy, but it is controlled eating up. For example, if you want to develop a hand, you know that a baby does not have 5 fingers. At the start the knuckles are like this, and slowly it will start breaking up from between, and fingers will start separating out like this, and it will form a complete hand. So what happened here is that from in between these fingers the cells was eaten out. So that function was actually by lysosome. So a lysosome actually caused the cell death of that specific cells that we didn't wanted. So that is called autolysis. Autolysis helps in developmental phase during your 9 months pregnancy and formation of baby and different body parts. So autolysis is programmed cell death, and that happens by the lysosome. There is another function of lysosome, that is exocytosis. Like for example, small exosomes forms like this, and it helps them going out of the cell. So this was about exocytosis. What about cell death? We already discussed about it. Like cell death is caused by lysosome. Lysosome causes eating up of all the organelles that are present, and lysosome is a suicidal bomb, and it can like cause the cell death. So you don't have to go into really deep depth of these functions. You only need to know the basics, like what is phagocytosis, what is autophagy, what is autolysis, what is programmed cell death, what is exocytosis. Like these are really important. So programmed cell death is actually autolysis. So these are the major 4 functions for lysosome. You have to know these major functions because they are really important. I wasn't going really deep into the topic because it is not that important if you go into the depth. These functions are really important and really relevant to your IMAT exam, but if you go into really deep, then these concepts will start becoming irrelevant. So this is the limit that you need to know, that I taught you. So you don't have to go any more deeper into the lysosome topic than we have just went now. Next thing, that the thing that we missed here is that there are diseases associated with lysosome. Maybe like 20 such diseases are present because that are caused by lysosome, and these are called lysosomal storage diseases. They are produced by mutation. For example, if you have less number of lysosomes, that causes the breakdown of glycogen. What will happen? Glycogen will start like filling up in your body, and glycogen level will spike, and you will have a lot amount of glycogen in your body. Then it will cause glycogenesis type 2. It will cause glycogenesis type 2 disease. It can also cause Tay-Sachs disease. So Tay-Sachs disease is a disease that happens because of accumulation of lipid. For example, if you don't have lysosomes that degrades lipids in your body, then you will have accumulation of lipid. A large amount of lipid will be accumulated, especially in your brain, and it can cause mental retardation, and like brain cells can die. So Tay-Sachs disease is a disease that is caused by accumulation of lysosome in your body. So glycogenesis is a disease that is caused by accumulation of glycogen, and Tay-Sachs disease is caused by the accumulation of lipids. Let me write it down into a box, and then you have Tay-Sachs disease. Glycogenesis is because of accumulation of what? Glycogen. And what is Tay-Sachs disease? It is due to the accumulation of what is Tay-Sachs disease? It is due to accumulation of lipids. So this was all about lysosomes. Next we will move to our next topic, that is about vacuoles. Like vacuoles is a small topic, we won't be going into very big detail. So vacuoles are like large vesicles that originate from endoplasmic reticulum that we just discussed, and they are like really present in both animals and plant cells, and the membrane separating the vacuoles is called as the tonoplast. If you see here, the vacuoles have a membrane, and the membrane of vacuole is called what? Tonoplast. So the membrane of vacuole is called tonoplast. If you talk about plant cell and animal cells, they have different kind of vacuoles. In plant cells, there is a large central vacuole which is present, that is found by like coalescence, or you can say like combination of many small vacuoles combining into one big large vacuole. When in animal cells, there are like so many vacuoles that are present throughout the animal cell. So that is the difference between animal cell and plant cell. Because the plant cell have a large vacuole, that's why the nucleus will be pushed aside towards the corner, and animal cell have nucleus in the center. We just discussed that in our nucleus topic. So we just revise the concept that nucleus is not in the center of plant cell, while animal cells have nucleus in the center of the cell. If we talk about the functions, so like vacuole is really important in animal cells. The food vacuoles are formed by phagocytosis, and many fresh protists have like contractile vacuoles. So animal cells have contractile vacuoles that helps to pump water out of the cell and maintaining the tonicity of the cell. In young plants, the vacuoles helps in protection of plants against herbivores, because they contains poisonous or unpleasant things to animals. So that's how plant cells can be saved through vacuoles, because it contains poison. If we talk about plant cells, the central vacuole also contains, in the center it is called as cell sap, just like your cytoplasm. So cell sap is present in the vacuole, and in plant cells it is contains inorganic ions or metabolic intermediates. Like they also expand the plant cell without diluting the cytoplasm. So vacuole also contains water storing abilities, so it stores a lot amount of water. So that's why they maintain the turgor pressure of water on the plant cells and animal cells. So they are like responsible for support and rigidity of the leaves and young parts of the plants. So this was all about vacuole functions and the small topic that we just discussed. So vacuole is helping in turgidity, in contractile vacuoles, in it contains poison, and it also helps in support and rigidity of the total cell. Next topic is about microbodies. These microbodies are also formed from endoplasmic reticulum. So if we talk about these type, they are also called microbodies. The first is peroxisome and the other is glycosomes. So peroxisomes has a primary function is that it breaks down long fatty acids, and it also detoxifies harmful substances and neutralizes toxic hydrogen peroxide. That is a major function. As you can see, hydrogen peroxide and peroxisome, the names matches the function. If we talk about glycosome, it converts lipids into carbohydrates during seed germination, so that's how you can get energy for the process. If we talk about enzymes, peroxisome contains oxidative enzymes that produce and degrade hydrogen peroxide, while glycosome contains oxidative enzymes that are specialized for glycosylate cycle, because glycoside cycle is the major cycle that happens in glycosomes. The main function that you have to memorize here is that peroxisome helps in hydrogen peroxide, right, and it helps in glycosylate cycle. If you talk about peroxisome, they are present almost in all eukaryotic cells, while glycosomes are present in only plants. If we talk about abundance, so peroxisomes are mostly abundant in liver and kidney cells. The reason is because liver and kidney cells contains detoxification function. So detoxification occurs in liver and kidneys. That's why they are mostly contains peroxisomes. While glycosomes are mostly present in sunflower, peanuts, and soya beans, because they are mostly helps in you can say storage of different oil seeds, and they require glycosylate cycle in the most earnest. And if we talk about pathways, so peroxisome has the function of neutralizing and detoxifications. That's why it is involved in the beta oxidation of fatty acid and photorespiration that happens in liver, while glycosomes has the pathway called glycosylate cycles that converts fats into sugars. The conversion of fats into sugar is called gluconeogenesis, and gluco means glucose and neogenesis means formations. So gluconeogenesis means formation of glucose. So that was all about peroxisome and glycosome. They are not really important for IMAT, but they are still relevant, so you should know about the basics of these peroxisome and glycosome topics. Next we talk about mitochondria. Mitochondria is like really important topic. You have to like go into like a deep detail, because mitochondria is the powerhouse of cell. You can also say it is the battery or the furnace of the cell. So these are the all the names for ribosome. You can also say it is battery of the cell, powerhouse of the cell, or you can also say it is the furnace of the cell. Why it is the furnace, you will know after we study about all these functions. So it is powerhouse, battery, or the furnace of the cell. So the first thing is that like mitochondria has different size and numbers. It can vary from 1 to 1000 per cell depending upon the activity of the cell, because mitochondria is abundant in your muscle cells. For example, if a person does a lot of exercise, then he will obviously have a large amount of mitochondria. But if a person does not work out a lot, then he will have small amount of mitochondria. So mitochondria size and numbers vary from cell to cell. If we talk about mitochondria, then the first thing that you need to know about mitochondria is that it is self-replicating. Why is it self-replicating? What does self-replicating means? Self-replicating means that it does not need any support from outside to survive on its own. Like it can survive on its own. No other organelle can survive on its own. For example, if you get mitochondria and separate it out from the whole cell, then it can survive on its own. It does not need the cell, but the cell need the mitochondria. So it is like a sigma, chad, you can say whatever it is, but mitochondria is not dependent on anything. It is self-replicating. And the other thing is that it contains its own DNA. It contains DNA. That's why it is self-replicating. It does not need anything else. It contains ribosomes. The first thing is that it contains DNA. The other thing is that it contains ribosome. We just studied that there were two types of ribosome, 70S and 80S. 80s ribosomes were present in eukaryotes, but we talked about that 70s ribosomes was also present in prokaryotes and eukaryotes. Where in eukaryotes? In eukaryote cells contains mitochondria, and mitochondria contains ribosomes, and the ribosomes in mitochondria are 70s. So technically eukaryote contains 70s and 80s both kind of ribosomes. 80s ribosomes are present in the cytoplasm and in the rough endoplasmic reticulum, while 70s ribosomes are present inside the mitochondria. So that's why they can also make their own protein. Like as you can see, mitochondria has its own DNA, it has its own information. Mitochondria has in its own ribosome. Mitochondria can make its own energy. So why does it need anyone else when it has everything else, right? As you can see, what is the prerequisite of life? It need to have DNA, it need to have protein, it needs energy. So mitochondria have everything. So that's why it is a self-replicating organelle. If we talk about the structure of mitochondria, so structure of mitochondria can be like, it can appear as a vesicle in the form of vesicles, it can appear in the form of like kind of rods, it can also appear in the form of filaments. So these are the appearances of mitochondria. It can vary from shape to shape. If we talk about the other structures, it contains two membranes, the outer membrane and an inner membrane. The outer membrane is smooth and it contains porins. Porins, you can say holes, and that's why the material can go in and out of the mitochondria. And first thing that you need to know is that these kind of porins are freely permeable. We talked about in a cell membrane that cell membrane is not freely permeable, it is semi-permeable. Cell membrane only cause one or two material to go inside, and it does not let everything goes in and out. So that's why plasma membrane is semi-permeable. It only permits few molecules to move in and out of the cell, while mitochondria is freely permeable. It does not stop anything from coming in and out for the smooth membrane. If we talk about the inner membrane, so inner membrane contains granular structures, and these granular structures are called F1 particles, F0, F0 and F1 particles, and they contains enzyme that is called ATPase enzyme, which are also called as ATP synthetase enzyme. So these are the particles that are present in the inner membrane. So inner membrane has kind of a rough appearance, and it contains these particles called F particles. In addition, like several other complexes are also present in the cristae of the mitochondria. What is cristae? Like just like you can see that this is the outer membrane, and you can see this is the inner membrane that is like giving you a velvety appearance, and that velvety appearance is actually called as cristae. You have to be really careful between cristae and cisternae. We discussed about cisternae in Golgi apparatus and endoplasmic reticulum, and these cisternaes were involved in endomembrane system, but this is different, it is cristae. The pronunciations are different, as I talked about, in different regions. Like some people say it is cristae, some people say it's cristae, and some people read it as cristae. I read it as cristae because it is much easier to memorize. You can pronounce it as you like. Okay, you can say it is cristae or whatever you want to. So the thing is that cristae contains velvety appearance, and cristae contains, first of all we discussed about, it contains F particles. It also contains electron transport chains. On those electron transport chains ATP is formed. We will discuss really in detail about electron transport chain in our chapter about respiration. In respiration we will discuss about how these ATP is formed in mitochondria. So the inner membrane divides the mitochondria into two compartments. The first is the inner compartment and the other is the outer compartment. So the first thing that you need to know is that the narrow region between like the inner membrane and the outer membrane, like this, as you can see this portion, this is a compartment, and this compartment is called intermembranous space. And then you have this internal compartment that I am highlighting, this one. Okay, So this is the internal compartment and it is called mitochondrial matrix, or you can simply say matrix, and it is enclosed by the inner membrane. So these are two compartments, the intermembranous space and matrix. So mitochondrial matrix contains everything that we discussed about. It contains the DNA, it contains RNA, it contains ribosomes, enzymes, coenzymes, salt, organic, inorganic ions. All of these things are present in the matrix, just like there is a cytoplasm of your cell. So let's talk about the table here. So there is an outer membrane that is smooth, permeable to small molecules, and it contains porins. And there is intermembranous space that is present between outer membrane and inner membrane. And intermembranous space is space between these two membranes, and it is the place where H positive ions accumulate. We will discuss about how H positive ion accumulates and why is there accumulation in intermembranous space in our topic for respiration. When we talk about electron transport chain, then this topic will be really clear. You just have to memorize these key processes. Then when we discuss these processes in respiration in detail, you will understand this in a better way. So just memorize these processes for now, and you will understand why these things are present in this membrane. Inner membrane contains electron transport chain and ATP synthetase enzyme that forms ATP. If we talk about matrix, matrix is the place which contains Krebs cycle and pyruvate decarboxylation and beta oxidation. These are the processes that happens in the matrix. So this was all about the four compartments for the mitochondrial structure: outer membrane, inner membrane, intermembranous space, and the matrix. So this was about mitochondria. If we talk about other things that like another MCQs that I want to discuss here is that where does the mitochondria in a person come from? Because we will discuss in our reproduction chapter, but let's discuss it here again. What is mitochondria? As you know, it is an organelle that helps in energy formation, right, in making an ATP. So where does the mitochondria come from? Does it come from the sperm cells, or does it come from the ovum or egg cells? Actually it comes from not sperms, but it actually comes from the ovum. So mitochondria comes from the mother. That's why females are really important. So actually the progeny moves from mother, not from father. Because there is a really big misconception in our society that the progeny moves from father to son to son to father, but actually the bloodline is kept actually by the mother, because most of the qualities that are present in the progeny is from the mother. Mother is the one that gives you the mitochondria, that gives you the athletic abilities, that gives you the most of the genetics to become a genius person. Obviously father has a really big influence, but mother has a greater influence in a person's future, because mother's genetics are stronger, because you are going to spend 9 months in your mother's womb. So that's why mitochondria comes from your ovum, not from the sperm. I thought that it is a important information, so I included that in here. So let's talk about our next organelle, that is plastid. We discussed about our first double membranous organelle, that was mitochondria. Oh, I just remembered a point. We talked about that there was a concept about envelope. So we don't say that mitochondria has an envelope, because mitochondria's inner membrane is not smooth. It has a velvety appearance. As you can see, the inner membrane has crusty present, and it is really velvety and does not have a definite shape. That's why we say that mitochondria is a double membranous organelle. We don't say that it contains any envelope, but we say that nucleus have an envelope. We say that chloroplast have an envelope, but we don't say that for mitochondria, because it has inner membrane that has crusty and you can say a velvety appearance. If we talk about next topic, that is about plastids. So if you haven't studied about plastids, then let me tell you what is a plastid. So plastid is an organelle that is only present in plants and algal cells. like they are necessary for essential processes like photosynthesis, food storage, and presence or absence of these pigments can like really influence a whole organism. So plastids are only present in plant cells. So first thing that plastid have is the proplastid. So the plastid that you have here is called proplastid. So proplastid will form different types of cells ahead. The different types of cells are chromoplast, chloroplast, leucoplast, amyloplast, elioplast, proteinoplast, and these are all the types of plastids that you will discuss here. So how these are formed, this is the structures that how it is formed. The first structure, as you can see here, is a proplastid. Proplastid will form first of all etioplast, and then it will also form leucoplast and chloroplast. From chloroplast you will form chromoplast, and from leucoplast you will have amyloplast, elioplast, and proteinoplast. So this was really important concept to know that how these organelles are formed. So if we talk about their functions, then their functions are really easy. You can just memorize them during the lecture. So first of all, we talk about these plasts that are from leucoplast, amyloplast, elioplast, and proteinoplast. Proteinoplast, the function of proteinoplast is simple. It stores proteins, as the name is telling. If we talk about elioplast, elio has L and lipid also has L. That's why elioplast helps in the storage of lipids. And if you have amyloplast, you know that amylose enzyme helps in the digestion of carbohydrates. That's why amyloplast helps in the storage of sugars like glucose and starch. These carbohydrates are stored in amyloplast. So amylo is came from amylose enzyme. Amylose enzyme digests carbohydrates. So amyloplast helps in the storage of carbohydrates. Amylo stores carbohydrates, elio stores lipid, and proteino stores proteins. If we talk about leucoplast, so leucoplast is the major branch. Like leucoplast is the type of plastid that helps in the storage, and it had three types we just discussed. We will discuss chloroplast later. First of all, we discussed about chromoplast. So chromoplast has a color, and chromoplast helps in pollination, like it gives colors to the leaves and petals of flowers and leaves, and chromoplast helps in pollination. So if we talk about etioplast, that is the last example, it helps in crystalline prolamellar bodies, and it is a precursor of chlorophyll. So these were the different plastids that were really important and you have to memorize. So if the main organelle, or you can say the main plastid that we have to discuss here, is obviously chloroplast. Chloroplast is the major organelle that is the most important and for our IMAT exam. So our next topic is about chloroplast, and chloroplast is the site of photosynthesis. It is the place where photosynthesis occurs. It is also called self-replicating because it contains its own DNA and its own RNA. It also contains 70 ribosomes. So just like mitochondria, it is self-replicating organelle. But there is a difference between chloroplast and mitochondria, and that is that mitochondria is present in all eukaryotic cells, while chloroplast is only present in plants and algae. Mitochondria and chloroplast both have own DNA, own ribosomes, and the difference is that mitochondria have two membranes, outer and inner, but the mitochondrial membrane has cristae and it has a velvety appearance, while chloroplast contains two membranes and both of them are smooth. That's why chloroplast and nucleus, these membranes are called nuclear envelope, and chloroplast has membrane envelope, while mitochondria is not called envelope because its membrane has a velvety appearance. If we talk about processes, mitochondria has aerobic respiration and chloroplast has photosynthesis. If we talk about ATP direction, mitochondria produces ATP while chloroplast uses ATP to produce glucose molecules. The key molecules in mitochondria are ATP synthetase, and in chloroplast is a chlorophyll molecule that is present on thylakoid membrane. So let's come back to chloroplast like we were discussing. So chloroplast, just like mitochondria, has outer and inner membrane, and it has intermembranous space that is present between outer and inner membrane, and it has thylakoid stroma too. If we see, like, look at the diagram of chloroplast, so the chloroplast has a inner structure and inner compartment that is called stroma, and then you have a intermembranous space that is present between outer and inner membrane. Mitochondria had inner compartment called matrix, and in matrix there was a process happening that was called as respiration, right? Calvin cycle was happening in chloroplast. So Calvin cycle happens in the stroma of chloroplast. And if you talk about what is other things that are present in stroma, and that is thylakoids. Thylakoids are like disc-like structure or cone-like structures that are present in your stroma, and there are two types of thylakoids. Thylakoids have two types. If we talk about thylakoids, it has two types. One is smaller thylakoid and the other is larger thylakoid. So there are two types of thylakoids. The disc-like thylakoid that you are seeing here is called as smaller thylakoid, and the thylakoid that connects between the stacks of thylakoids that you can see I'm highlighting here is a, what is it called? It is called larger thylakoid. So smaller thylakoids are disc-like structure and piled over one another, and if they form a stack, just like a stack of coins like this, and the whole stack, what is it called? It is called as granum. Granum is singular and grana is plural. So there are so many grana that are present in your chloroplast molecule. There are specific numbers associated with these. What are the specific numbers? If we talk about, these numbers are different, just like I talked before. The numbers could be different at Pearson, and then you have different numbers at Cambridge, and your different high school books have different numbers. So I wrote down the range. So there are 40 to 60 grana. This is the range of grana in chloroplast, and then you can have 20 to 50 thylakoids. So these are the numbers that you have to memorize, because these numbers can vary from book to book. So that's why I wrote down the range that they can fall between. These kind of MCQs are rare in IMAT exam because they don't ask about specific details, but they are really important because you have to know what are the number of granas and thylakoids, because they might ask. We don't know, we can't be sure about anything in competitive exams, so we have to be ready for anything that can happen there. The question could be straightforward, it could be about numbers, it could be about concept, it could be about process. So we have to be really careful and be really strategic while our preparation. So the next thing is that about the larger thylakoid. So the larger thylakoid actually connects, yes, you can see, both granas with each other, and they are also called as intergrana. So the larger thylakoid is also called as intergrana. So that was about small and large thylakoids. You have to know that photosynthesis does not occur on large thylakoid. The photosynthesis actually occurs on smaller thylakoids. So photosynthesis is the process that happens on smaller thylakoids. So these are the processes that are happening in chloroplast. You can see here that in stroma Calvin cycle happens. In double membrane, it is a place where independent origin, it is the process where endosymbiosis happens. We will discuss it in our evolution chapter. We'll discuss about Calvin cycle in our respiration chapter and in photosynthesis. And thylakoid is the place where photophosphorylation happens, and granum is the place, like it contains stack of flat membranes and it contains thylakoids. So we will discuss these processes in detail in our photosynthesis, in our respiration, and in evolution chapter. So these were all about the chloroplast. So chloroplast topic is also done. Let's move on to our next topic, that is about plant cell and animal cells. So this comparison is really important because I might have asked so many questions from this specific topic. So the table is really easy, to be honest. Animal cells does not contain cell wall. Cell wall is present in plant cells. Why is the reason? Because cell wall is actually rigid. Cell wall is really rigid and it is only present in plant cells. As you can see that trees and different plants are really rigid and hard. The reason is because of the cell wall. Cell wall is really hard and rigid, and that gives the appearance to the plant cells. If we talk about animal cells, then the cell wall is absent and animal cell contains only cell membrane. That's why animal cells are soft and they are squishy, and animal cells can do a motion. If animals also had cell walls, then the motion would have been impossible because the body would be so rigid to show any kind of motion. If we talk about chloroplasts are only present in plant cell because they have to do photosynthesis. If we talk about vacuole, animal cells have, like, does not contain central vacuole. The vacuole is at the peripheral region because they have small temporary vacuoles only. And if we talk about plant cells, then the plant cells contains a large single vacuole, and the large single vacuole pushes the nucleus towards the peripheral region, and that's why animal cells contains vacuoles in the peripheral region and nucleus in the center, while plant cell contains vacuole in the center and nucleus in the peripheral region. If we talk about centrioles, we already discussed it. Centrioles are only present in animal cells but absent in plant cells. If we talk about tonoplast is not present in animal cells because there is different small number of vacuoles, and the tonoplast is the outer membrane of a vacuole, while plant cell contains a large central vacuole. That's why it contains a tonoplast. If we talk about plasmodesmata, as actually plasmodesmata is a connection or a pathway between two plant cells. If we make it like this, this is one plant cell and this is an other plant cell. These two plant cells are connected with each other, and the connection is called as plasmodesmata. This channel or this pathway or this tunnel is called plasmodesmata. Plasmodesmata is not present in animal cells, it is only present in plant cells. If we talk about lysosomes, then animal cells have prominent and present lysosomes, while in plant cells lysosomes are not prominent. Actually, in plant cells, lysosomal function is carried on by the vacuoles. So vacuoles are the one that carry out the function of lysosome in plant cell. So vacuoles actually have digestive enzymes, digestive function in plant cells. So lysosomes are more prominent in animal cells, while in plant cells it is a controversial. In some cells lysosomes are present, in most of the plant cells it is not present. In most plant cells the function is went through by the vacuole. Vacuole also contain digestive enzyme, so vacuole can also do the function of lysosome in only plant cells, not in animals. If we talk about the shape, as you know that animal cells does not contain rigid cell wall, so that's why they are flexible, squishy, soft and irregular, while plant cells are rigid because of the cell wall and they maintain their shape. If we talk about animal cells, then their energy storage is glycogen, while in plant cell it is starch. So starch is the storage energy in plant cells and glycogen in animal cells. We discussed it in detail in our biological molecules chapter. We discussed about glycogen and starch in detail, and why animal cells contains glycogen and why plant cells contains starch. If we talk about mitochondria, it is not present in animal cells while it is present in plant cells. If we talk about ribosome is present in both animal cell and plant cell, but plant cell contains another ribosome, extra ribosome, that is 70S in chloroplast and 70S in mitochondria, while animal cell contains 80S ribosome and 70S ribosome only in mitochondria. So that was about animal cells and plant cells. So this table is really important. It might seem really easy to you. Most students already memorized this topic, but I might loves to ask questions from this table. Like, this table is like gold mine. So many MCQs come from this topic every year, so be really careful while you are solving the MCQs from this table. Let's summarize the whole chapter. These are the important functions and the membranes and different organelles. They are in front of you, all major organelles and their quick reference. You can take a screenshot, the notes will be available too. And then you have a DNA summary for all these organelles. Like DNA is present in nucleus, mitochondria and chloroplast, and other organelles does not contain any kind of DNA. If we talk about nucleus, it contains linear, while mitochondria and chloroplast contains circular DNA. If we talk about nucleus does not contain any ribosome, while mitochondria and chloroplast contains ribosome. If we talk about what kind of ribosome, that it is 70S. That's why nucleus is not self-replicating, because it does not contain ribosome. While it contains own DNA, obviously nucleus contains its own DNA, but it does not contain its ribosome, and nucleus cannot produce its own energy. That's why mitochondria and chloroplast have ribosome, they can produce their own energy, and that's why they are self-replicating. So that was all about your full chapter. We have completed our chapter number 4, and there is going to be a test. Complete that test, practice as many MCQs as you can, solve all the past papers, and you are good to go. Don't forget to solve your weekly mock full length papers. And this was all from my side. Keep working hard. Take care. Bye bye.